Hi. So I just want to go over some of the stuff I've been doing in class over the last week or so. A lot of you have missed out on it. So we were looking at the genetic crosses and the extra ones that are required at higher level. So first of all, we need to know here Mendel's first law, the law of segregation, and Mendel's second law, the law of independent assortment. So Mendel is known as the father of genetics because of the experiment work he did looking at genetic crosses in pea plants, and he came up with these two laws based on his experiments. So what I want to do is I want to look at a genetic cross where two traits are involved, sometimes called a dihybrid cross. So we're going to look at the example that would, some of the examples that Mendel would have looked at. So we'll say, for example, he studied pea plants, and one of the traits in pea plants was in relation to the seeds. And the seeds were either round or wrinkled. And yellow or green. And round was dominant over wrinkled, so therefore the gene for round will be a capital R, and the gene for wrinkled then will be a small r. Yellow is dominant to green, so the gene for yellow will be a capital Y, and the symbol we give for the allele for green is the small y. Remember, an allele is an alternative form of a gene. Now, so let's look at a cross between a homozygous dominant plant for both seed shape and color. So, that's crossed with a homozygous recessive plant. So the homozygous dominant, first parent, is going to be round yellow. It's homozygous dominant, homo meaning the same pair of alleles for a trait for each of these. And it's been crossed with the homozygous recessive. Now, so what we're working out here is what we call the F1, the first filial, or the first generation. So we have the parent and then the gametes from this. Now, going back to the the first law, Mendel's first law, the law of segregation. Traits are governed by pairs of factors. Mendel called them factors. We now know they're alleles that separate a gamete formation, with each gamete receiving one factor, or one of the alleles. So if we take the first trait, it's the seed shape, so the gametes can only have one of the pair. So then the only options we can have then in this plant, all the gametes will have the alleles capital R, capital Y. And in this plant, it's the same. It will have this, it can only have one set of gametes, and that's with the alleles small r and the alleles small y. There's no other combination that you can get from either of those two. So what happens, very straightforward then, the offspring, when during fertilization, when these gametes join, the offspring will be capital R, small r, capital Y, small y. That's the genotype. The phenotype then is all round yellow. Now, we're moving on to the F2, the second filial. We use the offspring from the F1 now as the parents. So here we have the heterozygous dominant plants from the heterozygous dominant plants from the offspring of the first cross now become the parents. Now look at the gametes. And in this case, we need to go to Mendel's second law, the law of independent assortment. So either member of a pair of alleles can combine with either member of another pair of alleles. So that means either of this pair can join with either of that pair. Okay, so we must look at all the different combinations. So we could have the capital R with capital Y, capital R with small y, small r with small y, or the small r with capital Y. And we see in this parent is the exact same. So all possible combinations. Now, so we can see there are 4 possible gametes in the first parent, 4 possible gametes in the second parent. So we're going to have a lot of combinations, possible combinations when fertilization occurs. So you could have first this gamete joining with any of these, that gamete joining with any of these, and so on. So really this is quite complicated. So we're going to do the Punnett square for this, just to make the matching up of the gametes or fertilization easier. So I take one parent and I put their gametes along the top. Sorry, I'm just going to swap these two around. The 2 recessive go at the end. And here's the other parents down along, the gametes from the other parent down along the side. Okay. Let me just draw lines across. Okay, so here we have our Punnett square. And now we match. We go across and down to find the matches. So here now we're working out the genotype of all the possible offspring. So I need to concentrate on this. Hopefully I don't make any mistakes. And we put the capital letters first in each case. We don't go first to the side or first from the top. We always go with the capital first. Okay, so that's our set of genotypes for this. Okay, now the next thing is to name the phenotypes based on each of these. So I'm just going to do a little bit of color coding here. So the first one up here, we can see, well, it's homozygous dominant, so that's obviously going to be round yellow. So let's see how many round yellows we have. So we'll just use a marker on this. So we see there, once we have a dominant R, or a capital R, and a capital Y, it's going to be round yellow. So that's 1, 2, 3, 4, 5, not this one, because there's two small Y's on that, 6, 7, 8, 9. So we have 9 round yellow plants. Right, the next one I'm going to look at is this one here, and we can see that that's has a capital R, so it's round, but it has two small y's, so it's round green. So let's count how many round greens we have. So we have that one. There's that one there. And I missed this one over here. Okay. So that's 3 round greens. So the next one then we are looking at is this one here. Okay. And we can see there's two small R's on that. So therefore it's wrinkled. But it has at least one capital Y. So therefore it's yellow. So it's wrinkled yellow. And let's see how many of these we have. So there's 1, 2, and 3. 3 of those as well. Leaving me then with the last one here, which is homozygous recessive. So therefore it's wrinkled. Wrinkled green, and there's just one of those. Now, so whenever you have a dihybrid cross where you have two traits together and both parents are heterozygous for each trait, you will always end up with this ratio. You will have all possible, the offspring will be all 4 possible combinations of the traits in the ratio of 9 is to 3 is to 3 is to 1. Okay. Now, so there are 2 other things at higher level genetics that we need to look at, and I'm going to make 2 other videos on those, and they are here in the higher level notes. The next thing we see there is gene linkage, and after that is sex linkage. Okay, so I will share those shortly. Thank you. Bye.